Hello, welcome to Biology, the scientific study of life. Cracking the code. Molecular biologists cracked the code of life in the early 1960s when a series of elegant experiments disclosed the amino acid translations of each of the RNA codons. The first codon was deciphered in 1961 by Marshall Nirenberg of the National Institutes of Health and his colleagues. Nirenberg synthesized an artificial mRNA by linking identical RNA nucleotides containing aerosols as their base. No matter where this message started or stopped, it could contain only one codon in repetition, U. Nirenberg added this poly-U to a test tube mixture containing amino acids, ribosomes, and the other components required for protein synthesis. His artificial system translated the poly-U into a polypeptide containing a single amino acid, phenylalanine, PHE, as we see here. Strung together as a long polyphenylalanine chain. Thus, Nirenberg determined that the mRNA codon UUU specifies the amino acid phenylalanine. Soon, the amino acid specified the codons AAA, GGG, and CCC were also determined. Although more elaborate techniques were required to decode mixed troubles such as AUA and CGA, all the 64 codons were deciphered by the mid-1960 years. As this figure seen here, what's that? The three termination codons marking the end of translation. Notice that the codon AUG has a dual function. It calls for the amino acid methionine, MET, and also functions as a start signal. Notice that. Or initiation codon. Genetic messages begin with mRNA codon AUG, which signals the protein synthesizing machinery to begin translating the mRNA at that location, because AUG also stands for methionine. Polypeptide chains begin with methionine when they are synthesized. However, an enzyme may subsequently remove this starter amino acid from the chain. First, mRNA base. Notice that. Also, in this figure, the dictionary of the genetic code. Notice it's clearer here. Like this. That there is redundancy in the genetic code, but no ambiguity. For example, although codons GAA and GAG both specify glutamic acid redundancy, neither of them ever specifies any other amino acid. No ambiguity. The redundancy in the code is not altogether random. In many cases, codons that are synonyms for a particular amino acid differ only in the third base of the triplet. Our ability to extract the intended message from a written language depends on reading the symbols in the correct grouping, that is, in the correct reading frame. Consider that statement: the red dog ate the cat. The red dog, between two brackets, the red dog ate the cat. group the letters incorrectly by starting at the wrong point, and the result will probably be gibberish. For example, here add, notice that. Here added oga, tit, hick, at. The reading frame is also important in the molecular and language of cells. The short stretch of the polypeptide shown in this figure, notice that. Well, transcription, translation, notice that the DNA strand template and mRNA or messenger mRNA protein. For instance, will only be made correctly if the mRNA nucleotides are read from left to right, 5 to 3, 5 to 3, or 3 to 5. In the groups of the three shown in the figure, U, G, G, U, 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 G, G, C, U, C, A, although a genetic message is written, although no space between the codons, the cell's protein synthesizing machine reads the message as a series of non-overlapping three-letter words. The message is not read as a series of overlapping words, U, G, G, U, U, notice that, and so on, which would convey a very different message. Evolution of the genetic code. The genetic code is nearly universal, shared by organisms from the simplest bacteria on the most complex animals. The RNA codon CCG, for instance, and is translated as the amino acid proline in all organisms whose genetic code has been examined. In laboratory experiments, genes can be transcribed and translated after being transplanted from one species to another. Bacteria can be programmed by the insertion of human genes to synthesize certain human proteins for medical use. Such applications have produced many exciting developments in biotechnology. Exceptions to the universality of the genetic code include translation systems where a few codons differ from the standard ones. Slight variations in the genetic code exist in certain unicellular eukaryotes, and in the original genes of some species. Some prokaryotes can translate stop codons into one of two amino acids not found in most organisms. Despite these exceptions, the evolutionary significance of the code's near universality is clear. A language shared by all living things must have been operating very early in the history of life, early enough to be present in the common ancestors of all modern organisms. A shared genetic vocabulary is a reminder of the kinship that bonds all life on earth. Notice that a tobacco plant expressing a firefly gene. Notice that. Back to second mRNA base, also the dictionary of the genetic code, the three bases of an mRNA codon are designated here as the first, second, and notice that, and third bases reading in the 5 to 3. Notice that 5 to 3. Direction along the mRNA practice. Notice here the codon AUG. Notice that AUG here. Only stands for the amino acid methionine, but also functions as a start signal for ribosomes to begin translating the mRNA at that point. 3 of the 64 codons function as stop signals, marking the end of the genetic message. Notice that. Okay, thanks for listening.